Part of the Canadian Society for Chronobiology, we have a special distinguished talk. This is like, what is the ruler thing? They have at SRBR almost. Yeah. This is our very, very distinguished talk. And I'm going to turn it over to Nico for a minute to explain why this one.
Thank you, Tammy. So since I'm a bit like the memory of the society, Tammy has asked me to say what is this lecture, so I won't take more than like 45 minutes to explain to you what's the Mrosovsky. Even no, less than 4 to 5 minutes. Joking. Okay, so as Tammy said, the Mrosovsky lecture, the Nicholas Mrosovsky lecture, is our big keynote lecture at our meeting, and this since the very beginning of our society 10 years ago in 2013. And I won't take like the time it would deserve to like describe all the things that Nicholas Mrosovsky has done, but he's really one of the founder, one of the pioneers of chronobiology in Canada. He's very well known for his work on non-photic resetting of clocks, his work on masking, for example, masking by light of locomotor activity in rodents, for example. So he has done seminal work, he has collaborated with many people in the field. So it was really natural for us when we started the society 10 years ago to acknowledge the contribution of Nicholas Mrosovsky to chronobiology in Canada by naming our main keynote lecture after his name. Nicholas was really a particular person. Those of you who have known him know the kind of character he was, and he was a great person. I remember on a personal note the sharing, like I was at a meeting in 2001, and then I met him, and then he looked at my name tag like that, and he told me, "Do I know you?" And then we got to present ourselves. And then the year after, in 2002, which was my first SRBR meeting, I shared the room with him. I had the time to have good time with him, discussing with him. I learned at that time that he had this project on circadian rhythms, but also another project on sea turtles. He was regularly going in the Caribbean to study turtles. So so this is interesting, and this shows his wide interest. So we decided to have the keynote after his name. It was in 2013. As you can see here, he passed away 2 years later, which means that he knew about the honor that was made to him of naming our keynote lecture after his name. At the 2013 meeting, there was even a message from him. He was not able, he was already quite ill. He was not able to attend, even if it was in Toronto, but I think there was a recorded message from Nicholas that was broadcasted in the room there, so it was really moving. So I know he was honored by that, and we've continued since then, and we always want to invite somebody who would not necessarily doing similar work to what Nicholas was doing, but either someone who had been in contact with or who carries this ideal of going in uncharted territories, doing new kind of work and so on. So, and if I don't have more time than that, but if you're interested to knowing more about Nicholas Mrosovsky, Pat Lakin-Thomas had put together, with the help of many people who knew him, this fantastic video that we have on the website of the Canadian Society for Chronobiology. So go to the events page, and then you scroll down to the end, and then you'll see that video. I don't remember how long it is, but it's a good watch. So I really encourage you, especially those younger researchers among you. It's really an enlightening video. So with this said, now I will turn to you. I thought to Dennis.
Yeah.
So thank you very much.
Okay, so we've asked 3 people in particular to do double duty at this conference, and we have a keynote speaker from the cardiovascular conference who also shared her science with us. We have Sachin, who did public and shared his science. And then Frank, you saw a little bit yesterday, but this is the keynote that he came for today. Thank you to all of you guys for doing that. Really appreciate it. I'm going to have Dennis, who really helped me put this conference together, and I could not possibly have gotten all this organized without him, to do the honor of introducing Dr. Frank Scheer.
Thank you. Thank you. It is my great pleasure to introduce Dr. Frank Scheer. Dr. Scheer is a professor of medicine at Harvard Medical School and a senior neuroscientist at Brigham and Women's Hospital. Dr. Scheer co-founded and currently directs the Medical Chronobiology Program, an interdisciplinary research program at Brigham and Women's Hospital to foster translational research in sleep and circadian biology to understand the basis behind time-variant changes in disease severity, the adverse cardiometabolic effects of circadian misalignment, the effect of food timing on glucose metabolism and energy balance, and their interactions with genetic variants. Understanding the biological basis of these changes across the day and night and their interaction with genetics promises to help in the development of personalized and time-based behavioral and pharmaceutical interventions, such as appropriately timed meals, exercise, light exposure, melatonin intake to target the body clocks to prevent, treat, and manage diseases, including hypertension, asthma, obesity, and diabetes. Thank you for joining us, Dr. Scheer. I muted it, so I don't make noise.
Thank you so much for that kind introduction, and I can't say how honored I am for this distinguished honor to give this most esteemed lecture, the Nicholas Mrosovsky Lecture. So I hope I can live up to the high expectations that I'm sure all of you have at this moment. But I'm really delighted to be here, and I would like to thank Tommy and the rest of the organizing committee and congratulate the Society on your 10-year anniversary, which I understand it is. So I do also want to point out what Nico already so greatly explained, the enormous contributions that Nicholas Mrosovsky has had to the circadian field specifically, but also beyond. So, born in Romania to British parents, he then studied in the UK at the University of Cambridge, went to the University of London, and then made a big trip across the Atlantic. And so in that way, I kind of followed in his footsteps, I guess. But then stayed and he had a triple appointment in the departments of zoology, psychology, and physiology at the University of Toronto. So during his long active career, he touched on many aspects of circadian biology, but he really started off to study hibernation. So he also, like many of us, entered into circadian biology from other fields. But he has made tremendous contributions, as was already mentioned by Nico, to the understanding of non-photic entrainment, to the understanding of masking effects. So effects, for example, of light that are not due to shifting of the clock, but acutely. And the most well-known of, obviously, is the acute suppression of melatonin, but there are many others. And he actually did a lot of work in frogs and turtles, and the turtle work actually led to him to devote a large part of his career to conservation. Although he said he kind of dabbled in that, but I think he made tremendous contributions to that. He's also published enormous amounts. I think his first paper was in Nature, so good, off to a good start. Published, I think, 4 books. I just list 2 here. Got many awards. Organized the Gordon Conference on Chronobiology in 2001, and so and most important, I think, for this meeting and for this symposium and this lecture is the establishment in 2013 at the first meeting of the Society, the keynote lecture in his name. And so, but I want to also point out that actually tomorrow happens to be World Sea Turtle Day, and so I think it couldn't have been timed, well, maybe one day later would have been better. Couldn't have been timed much better than this. So thank you for the honor. So here's a brief overview of what I hope to do today. I first want to talk about energy balance, and I think this lines up a little bit with Nicholas Mrosovsky's work in metabolism and energy control. Then I want to talk about specifically more glucose control, both of which related to meal timing and glucose control specifically talking about interaction with genetics. Then talking about circadian misalignment effects and the effect specifically on cardiometabolic function, as well as the question whether we can implement any changes to help shift workers and to help in fighting the chronic disease risks. So meal timing and energy balance control, of course, relevant to weight regulation. And so when I was preparing some of these slides, some time ago, my wife called from the other room. She said, "Oh, Frank, have you heard about this new study related to body weight and health?" And so this study showed that women, well, you can read it yourself, so I thought I have to share this. I have to share this with you. But really, on a more serious note, the real interest in meal timing, and of course it's not difficult to get a chronobiologist interested in meal timing, but the real interest started with the observation by Diane Arbel in Fred Turek's lab, where the study approach was simple, but the consequences were profound. And so, surprisingly, nobody had really looked at this question in this way. And what they did is they used a high-fat diet, which is the most commonly used obese model in the field. And what they simply did is either provide the food access only during the inactive phase, during the light phase, or the active phase, the dark phase. And found this profound effect that the obesity development was more profound with mistimed food, so timing of food during the inactive phase, the light phase. So then in 2012 I met Marta Garolet, and I will show a picture of her in a second, when she was visiting Boston, and we got to talk, and we shared our common interest in understanding whether this actually may apply to humans as well. And so this was a collaboration that is now over 10 years old and has been very successful, pleasant, and influenced my work to a great deal. And so what Marta had is she had this unique database of over 400 obese and overweight men and women who had undergone a standardized weight loss approach over 20 weeks. But the question we wanted to ask is there a relationship between when people choose to eat and how successful they actually are? Because some people are more successful in weight loss than others. And what we found was indeed there were big differences. If we did a median split analysis and looked at the people who ate after 3 p.m. in Spain, lunch is the biggest meal, 40% of the daily caloric intake, versus those who eat earlier or later. So the people who chose to eat their main meal early lost about 30% more. Now, this was an observational longitudinal study. We didn't prescribe food timing, and so really, this is just that. It's an association. So, but we looked at whether there were differences in caloric intake or sleep duration or physical activity, self-report, etc., and to our surprise, none of these could explain this observation. But when we published this, somebody showed a cartoon earlier. I agree. I mean, when a cartoon comes out based on your work, I think then you've made it, right? So anyway, so very funny. So as he tries to cram in as much food as he can before 3 p.m. so he doesn't get fat. So that should not be the take-home message here. So this meal timing here was based on clock time, the clock on the wall. But that's not what we're talking about when we're talking about chronobiology. We're talking about, or at least most of us are thinking about the biology that actually underlies this. And so one gold standard circadian phase marker in the field is looking at the dim light melatonin onset. So when does your melatonin start to rise in the evening hours? And when do you eat relative to this central circadian time? So this was the question that Andrew McHill asked, who is now an assistant professor at Portland, Oregon at OHSU, where he studied over 100 college students and asked them to follow their eating pattern for a full week by using a phone app to take pictures of each and every single food item that then was analyzed for nutrient content. So he used that data for each individual, and then he compared this to the caloric midpoint, so when did they eat 50% of their daily calories, relative to the timing of their central clock. So dim light melatonin onset is the dotted line here in these two examples, and the solid arrow is the caloric midpoint. And what he found, and these are just two examples, is that the people who ate their calories earlier, so longer before their dim light melatonin onset, were on average had a lower body mass index and had lower adiposity. And I think interesting here also to comment is that when instead of using circadian phase of eating, he used just time of day of eating, so clock on the wall, the association fell apart. So suggesting that actually the circadian phase of food consumption may be important. Now, doing a dim light melatonin onset is involved, it requires skills, it's expensive, and it's not easily scalable. So another question is, can we get to a proxy measure of circadian phase of eating? So this was the question that Xiao asked, who's assistant professor at University of Texas, where instead of using DILMO, she used eating time relative to the individual's habitual wake time and bedtime as such a proxy. In addition to also look into this question, she also looked at whether there was a relationship with the chronotype of the individual. So what did she find? Well, she looked at over 800 participants who collected the food diaries that included timing of food over a 1 year segment. And what she found was that the individuals who were in the highest quintile of eating in the morning had reduced odds for having obesity or overweight, and vice versa. Those who ate in the last 2 hours in the highest quintile had an increased risk or odds ratio. And this was even more extreme in those dependent on their chronotype. So if those were morning chronotype people, they were even at decreased risk, further decreased risk when eating in the highest quintile in the morning, and vice versa for the late chronotypes. So what it suggests is, and this makes sense, right, if you think about it, so sleep is gating the information of light in most of us, in the sense that we're putting ourselves in a dark environment, we close our eyes. So it's not that sleep drives your clock, to some degree, a little bit it does, but primarily it's gating of light. And also here she found that the association fell apart when using clock time instead of time relative to the sleep-wake cycle. So subsequently, many labs throughout the world have investigated the importance of meal timing, and even though there is some controversy, there is a general consensus that late eating is related to increased odds or risk for obesity and overweight. So the question is then, why is that, right? So because a calorie is a calorie, so why would it matter when you eat it? Well, if you think about it, a calorie is still a calorie, but how your body interacts with it and how it's received by your body may differ. And I'll explain this in the next few slides. So this was the question we had. So what is the effect truly of experimentally shifting the eating fasting cycle without changing sleep-wake, without changing light-dark cycle, without changing physical activity, posture, all those other things, and without changing the amount of food or the composition of the food or the fasting duration. So we wanted to know, might it be mechanism involved in increased drive for food intake or a decrease in the expenditure of calories, so do you burn fewer calories, or might it have something to do with changes in, let's say, adipose tissue as your primary storage organ that may help explain these observations. So in order to test this question, so Nina Vujovic, during her postdoc in my lab, conducted this randomized crossover trial where people were randomized to either enter into the protocol for the early eating condition, and then after a washout of a few weeks into the late eating condition, or vice versa. So what I'm showing here on the left is, if you look at the two bottom ones, those are the middle one is the early eating condition and the bottom one is the late eating condition. And as you can see, the only difference they have is the timing of food intake, which is the blue rectangles. So the meals were equispaced, so they were 4 hours and 10 minutes apart in all the protocols. And so really what we did is we just delayed in the late eating condition by 4 hours and 10 minutes the first meal. And so what we did on the third and the sixth day, we had intensive measurements of energy expenditure by interaclometry repeatedly across the waking episode, hunger and appetite scales, leptin and ghrelin assessments, measurements of core temperature continuously. And then on day 5, we took a abdominal subcutaneous adipose biopsy in each of the both protocols after a fixed fasting duration in both cases. And so the other thing we did is that 3 days before they came into each of the in-lab conditions, we gave them all the food that they had to eat in particular time windows. And for 3 weeks before each in-laboratory visit, they also maintained a fixed sleep-wake cycle. So as best as we could, we tried to clamp down all the other factors so that we could purely look at the effect of changing of the mealtime. And the fact that we looked on day 3 and 6 is so that we can look at both acute and effects to see if it was these effects are maintained. What did we find? Well, let's first look at drive for food intake. And so here's indicated the hourly assessment or reporting by the participants using a visual analog scale on a computer each hour to indicate how hungry they were. And we have also many appetite measures. And what you can see is that in the late eating condition, which is shown in red, they are substantially more hungry, especially in the early part of the day, and it doesn't really compensate towards the end. And indeed, if you look, and there was no interaction between the two test days, so we collapsed the data, and so we looked at the probability for individuals to report being hungry, defined here as a visual analog scale rating of over 50%. And what we found was that the late eating compared to the early eating within the same individuals resulted in a significant increase in hunger. So what about leptin and ghrelin? Well, leptin and ghrelin actually were consistent in that regard, that leptin levels decreased and the ghrelin to leptin ratio increased. So leptin is a satiety hormone, ghrelin is a hunger hormone. We looked at acylated ghrelin, so the active form of ghrelin. And what you can see, for example, the bottom graph, the ratio, you can see on the right, the bar graphs, that across the wake episode, the ratio was increased with late eating. It was decreased with across sleep, but then across the full 24 hours, it was significantly increased. So the results are consistent between both the subjective and the objective measure, indicating an increased drive for food intake with late eating. What about energy expenditure? Well, the data indicate a decrease of energy expenditure. So on the top is indicated interaclometry measured every 1 or 2 hours. We measured it whether or not they were consuming the meal, and when they were consuming or not consuming the meal, they were maintaining the same semi-recumbent body posture, so to make sure that the physical activity levels were the same. And so what you can see is that, again, on the right bar graph, you can see that across the waking episode, there's a significant decrease in energy expenditure in these individuals. The downside of the approach we use, which is a ventilated hood within the raclometry, is we can't assess energy expenditure during the sleep episode. So what we did is we looked at core body temperature, which is shown in the bottom panels, and as a kind of poor man's proxy, you could say, of energy expenditure, because the physical activity was the same, the environmental temperature was the same, and so heat dissipation would probably be not a primary driver of changes in core temperature. Anyway, if we look at the waking episode, indeed, when we see a decrease in energy expenditure, you also see a decrease in core temperature. And when we look at the sleep episode, we don't see a compensation. There's not an increased temperature at night. In fact, the last 4 hours, there's a decrease in core temperature. So across the 24 hours, core temperature was decreased. So based on these two metrics, again, consistent results indicating a decrease in energy expenditure with late eating. What about the adipose biopsies? Well, this was really thanks to Matthew Brady, who's the brains behind this, to look at the transcriptomic analysis, indicating that with the biopsy taken in the individuals when they were in the late eating protocol showed increases in pathways for adipogenesis and decreases in those for lipolysis. So these two pathways, again, consistent with the notion that the fat would be in a state more likely to grow. And so while we set out to test which of the three mechanisms might be involved, to our surprise, we actually found evidence for all three mechanisms moving in the same direction, suggesting that late eating indeed would, at least in the short-term study, providing evidence that these pathways may be involved, these mechanisms may be involved in an increased risk for obesity. Now, the main limitation, obviously, of this study is the short-term duration, but it was really meant to test the potential mechanisms, the drivers that might underlie this. Now, then the question is, why might this be? Why would timing of food intake matter? So we had some previous data which suggested that maybe one of the factors that may be playing a role is the observation that diet-induced thermogenesis, also known as the thermic effect of food, which is the increase in the calories you burn after the consumption of a meal, that this is under circadian control. And so what we found was that the amount of calories after an identical meal in the morning versus the evening in the morning hours is much higher, such that if you would eat most of your food later, theoretically this could result in a reduction in the amount of calories you burn, and so theoretically could contribute to increased risk for increase in body mass with late eating. So the other piece of data we found and then was replicated by many labs, and I've shown here a few on this slide, is that hunger and appetite are under circadian control. So hunger and appetite is not only driven by homeostatic factors, it's also driven by your circadian system. And so this was assessed in the forced synchrony protocol, where we could assess with visual analog scale, and we could dissociate the role of the circadian system from the role of the fasting duration and the sleep-wake cycle, etc. So, and then more recently, Jingyi Cheng has shown that also acylated ghrelin is higher during the biological evening, which would go hand in hand with hunger being higher at that time, and also that has been shown by other groups as well. So it may be that these two factors play a role. So circadian control of these, of both the arms of your balance, if you like. Okay, so what about glucose control and the importance of timing? And so we and others have done a lot of studies to look at to what degree the well-known decrease in glucose tolerance from morning to evening to what degree that's due to the circadian system or not. And so we've used various protocols that I won't have time to go into today, but basically what we've shown is that the circadian system plays an important role and so we have a better glucose tolerance, meaning that if we are exposed to an identical glucose load, we can bring down the blood sugar levels much more efficiently. And in part, and we heard yesterday a beautiful talk about the pancreatic beta cells, and indeed that's what we find as well in vivo human studies. But in addition to, or maybe as one of the arms or the effector systems of the circadian system that we don't think is the whole story, but one of these arms might be melatonin that is influencing glucose control. Now, the interest in the field of the diabetic field for melatonin and circadian biology really, I think, was kickstarted by the discovery in the late 2008 and 2009, where the MTNR1B risk variant was discovered. So the melatonin, one of the two high-affinity melatonin receptors, so MEL1B or MT2, depending on which system you like, it's the same thing, is associated with increased risk for diabetes, increased fasting levels. Now this was published in 3 papers in the same journal of Nature Genetics in 2009, basically all based on primarily GWAS, genome-wide association studies, supplemented by additional evidence. And so really this was firmly planted as a very solid finding. And so the interest in melatonin regulation of glucose really took off there. Now this variant, why might this be important? Well, one of the reasons is that actually the variant is quite common. So it's so common that if you take both homozygous and heterozygous individuals, about half of the room here will be a carrier, as am I. So we wanted to know whether these observations that were done in GWAS, whether they may be looked at in a different way. And I'm saying this is a bit convoluted, but so think about it. In the GWAS studies, most of the observations on glucose control were based on daytime assessments. So because these are based on clinic assessments, well, most of you will know that during the daytime there's not much melatonin in the circulation to speak of. So we wanted to know if you now look at people who are carriers and actually elevate melatonin, may you actually see a more pronounced effect on glucose regulation. So this is what we did, and so the first study, and this was together with Marta Garolet and Richa Saxena. Richa is a geneticist from MGH in Boston. We've been collaborating since about 2009, a fabulous collaboration. I'm very lucky to be working with Richa. And so together we then asked the question whether if we administer melatonin orally, 5 milligrams, which you can just buy, no, in Canada. Can you buy this over the counter? Yes. Okay, just like in the US. So 5 milligrams of melatonin, which, as you probably will know, shoots up your melatonin levels to easily 100-fold higher than your nighttime levels. So pharmacological concentrations. And so we found that indeed with pharmacological concentrations, both in the morning and the evening, glucose tolerance is impaired. We then also wanted to know, well, is this just because it's a very high dose? What about physiological concentrations? So the approach there was to look at early or late dinner timing. So either 4 hours or 1 hour before habitual bedtime, when either melatonin levels are low or elevated. And again, what we found there is that glucose tolerance is impaired with late eating, and both of these effects were more extreme in the carriers. So, and at the bottom I'm just showing a conceptual paper that we have brought out for those of you who are interested. But more recently, last year, with Marta and Richa, and others, we published this work in Diabetes care where over 800 participants in Spain were exposed to an oral glucose tolerance test either 1 or 4 hours before bedtime. The late eating are the black curves on the left, and the early eating is the gray curves. And then from the top is glucose, the bottom is insulin, the left is non-carriers, heterozygous carriers, and homozygous carriers. And on the right, the bar graphs, you can see the dose-response relationship dependent on the number of alleles you carry for this risk variant, with worsening glucose tolerance and decreased pancreatic beta cell function. So based on these observations, we would, we promote people to think about the potential adverse consequences of either eating late or taking melatonin close to a mealtime for their glucose control. So then we wanted to I want to talk briefly about circadian misalignment, and we've heard a lot about this today, so I really enjoyed our lunch discussion today as well about model systems and shift work. And of course, and this has been shown over and over, so I don't need to dwell on this, but about 14% or so of the US workforce, and similar in Canada and many other countries, are engaged in real night work. So either permanent night work or rotating shifts with substantial night work component in it. And as we've heard as well, this is associated, based on epidemiological work, on increased risk for a lot of cardiometabolic diseases, but also cancer, asthma, depression, and the list goes on and on. Now, the epidemiological work has tried to correct or adjust for factors that may help explain the increased risk for these chronic diseases, such as socioeconomic status or lifestyle or workload. And so what they have found is that these factors cannot fully explain the increased risk. So what is it about circadian misalignment? What is it about shift work that might cause this? And circadian misalignment is one of the ideas that has been tested in the field. And in fact, if we look at the list of consequences of experimental circadian misalignment, if you use a slam shift in a laboratory or you use a force synchrony protocol, you can induce. We've spoken about what kind of models should we use and protocols we should use. It doesn't really matter what type of protocols one uses. These lead to worsening of glucose tolerance. We've shown that this might be due primarily to muscle insulin insensitivity, changing energy balance control, blood pressure, immune system, mood, and microbiota changes. So just very quickly to show one recent paper that was really a collaboration with Rush University, so led by Ali Kesevarjan's group, and Marta was included as well, in which it was really a proof of principle study to see first, is the oral microbiota in humans rhythmic, independent of the sleep-wake, rest, activity, feeding, fasting, light-dark cycle. And why oral microbiota? Well, stool is difficult to sample repeatedly across the day, whereas saliva is quite easy to do that. So what did we find? Just very briefly, so these are just some examples of microbiota populations that show a true endogenous circadian rhythmicity as assessed in concert routine conditions, so where we take out the sleep-wake cycle, rest, activity, posture, light, dark, etc. So we did this before misalignment and then repeated the same, so a second concert routine after misalignment, and what we found was actually that a number of these rhythmic population structures were no longer rhythmic, so an after effect of circadian misalignment. Not only were there differences in rhythm, there were also differences in population structure per se, so average across the 24 hours of repeated assessments, so we assessed it every 4 hours. And so what this suggested, and so this was done on 16S, so this is somewhat of a limit, although we know for sure that these are bacterial. Using inferred pathway analysis, it suggests that these factors that are changed relate primarily to metabolism and immunity. So it's not just your healthy controls, unfortunately, that are affected by circadian misalignment. Chronic shift workers, when brought into the lab and compared between circadian alignment and misalignment, also show impairments, meaning increases in high-sensitive COP, increases in blood pressure, impairments of glucose tolerance, etc. And so this is work that's in press, which was led by Jingyi Cheng, who's now a junior faculty in my group and going up for promotion to assistant professor, who looked in chronic shift workers at the regulation of acylated ghrelin, both in circadian alignment and circadian misalignment, so blue alignment and red misalignment in both these protocols, several weeks apart. And what she found was that acylated ghrelin levels, when misaligned, resulted in increases in acylated ghrelin across the 24 hours, but it was really driven by the increase towards the end of the scheduled sleep episode and surrounding the breakfast. Now, this may seem surprising unless you start to think again about the fact that ghrelin is under circadian control and the fact that breakfast, when misaligned, is actually happening in the evening hours when the circadian system increases ghrelin. So what we're really looking at is an interaction effect of circadian misalignment and a circadian phase effect on ghrelin. And so one obvious thought would be, well, maybe this is all due to the disruption of sleep, because it's true, when you sleep during the day, you don't sleep as well as when you sleep at night. So we measured sleep by polysomnography. We included it into the statistical model, which showed us that the effects observed are above and beyond any effects on sleep. And in fact, very elegant work by F. and Counter's lab experimentally confirmed that indeed these effects are not just explained by sleep. And so this really raises the question, if it's not the effect through sleep, might it be that it's actually the misalignment of the eating fasting cycle that is driving many of these effects we see? So this was really the motivation for the next study. So the question was, can we prevent the adverse effects of shift work or simulated shift work or circadian misalignment with meal timing when we fixed meal timing to the biological day, the circadian day? And this really is based on the animal experimental beautiful work by A.J.L and colleagues already now 23 years ago, where they put animals, nocturnal rodents, on either a nighttime feeding protocol when they would normally like to eat, or they put them on a daytime feeding protocol. And what they found is that the liver clock, as many of you will know, can be fully shifted in just a couple of days, fully shifted by about 12 hours if you move the eating window by 12 hours. So clearly the liver clock can be shifted by food timing. What happens to the SCN? Nothing. So the SCN doesn't really care in this protocol where they were kept under a light-dark cycle. And so what you end up with is a situation of internal circadian misalignment. So different clocks are running on different time zones. So we wanted to see if we could get some answers in human experimental protocols. So this is work that Sarah Shalapa led just last week. Sarah WhatsApped me that she now just started her associate professor position at Southampton University in the UK. So I'm really proud of her, as well as all the other collaborators, of course. So what Sarah did is she randomized people to either the control protocol, which is on top, or the intervention protocol, which is at the bottom. Now, it's a slightly difficult protocol, complicated protocol, but the essence of it is that we had two concert routine protocols at the beginning and the end, and in the middle we had a force asynchrony protocol to maximally misalign individuals. Why did we do this? Well, we wanted to assess with the concert routine protocols, again, just to recap, so we keep them awake, we keep them in dim light, we give them isocaloric snacks every hour, and they remain in the same constant body posture semi-recumbent. So we remove all the rhythmicities of behavior and environmental factors, and so we can extract endogenous circadian control. So we did this before and after in the orange bars, and then the force asynchrony is from day 7 through 11, in dim light, both of these protocols. And so the hypothesis we had is, well, maybe it's that we know that the central clock is not entrained during the force asynchrony at 28 hours. It's outside of the range of entrainment. You've heard this term before. So we know the central clock ticks along with a rhythm of about 24 hours, but might it be that some peripheral oscillators actually are entrained to our 28-hour force asynchrony protocol, where the sleep-wake cycle, rest activity, eating-fasting cycle are on 28 hours? And if so, might we see some remnants, might we see some circadian rhythms that have slightly shifted in the second concert routine protocol? And if so, can we prevent this change by the intervention, and the intervention being that we lock down the eating moments to exactly 24 hours. So it's shown in the bottom protocol. So the light blue boxes indicate mixed meal test protocols, where we had frequently sampling of glucose and insulin, and I'll show you some of those results, both aligned and misaligned, and we could look at circadian phase as well. And the red boxes are all the other meals, and all the meals were calculated, they had to eat all the food we gave them, etc. And the blue boxes are 10% of the daily caloric intake, those are snacks. Okay, so what did we find based on circadian markers comparing the first and the second concert routine? I'm going to show orange on the left for the control protocol, and blue I'm going to show the intervention protocol. Black is the baseline, and the colors are after. So the black is the first concert routine, and the colors are the second concert routine. So we've used core body temperature in the concert routine. This is a gold standard circadian phase marker. You can't use it in the regular sleep-wake cycle, but in the concert routine, it's a gold standard circadian phase marker. And what we find is that, as we expected, it doesn't do much. The misalignment, either in the control or the intervention protocol, doesn't really shift or change the rhythm of core body temperature. It may seem like the amplitude is a bit smaller in the intervention protocol. That's not significant. However, to our big surprise, I mean, we expected we may see a slight shift in the endogenous circadian control of glucose, but what we found was almost an inversion of the protocol of the endogenous circadian rhythm in glucose. In the control protocol, and we could fully prevent this occurring in the intervention protocol. So if you want to look at it in a different way, and many of you are used to this, if you look at it on a radial plot, we can see, and what's displayed is the delta. So it's the difference between the baseline, so the first CR, and the after misalignment CR. You can see the dotted arrow is core body temperature, so it kind of drifts across the protocol by about half an hour in both cases. But the endogenous circadian glucose rhythm is completely out of whack on the control protocol and recovered in the intervention protocol. Now, what about glucose control? Because this is fun for this audience, but what about insights into risk for health and for diabetes and glucose control? So in the control protocol, when people were misaligned, as we expected and had found before as well, the glucose tolerance is impaired. So from baseline in black to orange is in the misaligned. So black is aligned on the first force asynchrony day. Orange is misaligned on the fourth force asynchrony day, when they're 12 hours out of phase. You can see the impairment of glucose tolerance readily. And if you look at insulin, you can also see that the increase in insulin is sluggish. It's slow to rise despite the fact that the pancreatic beta cells see the same concentration of glucose. Because if you look at the first 40 minutes, you can see that the glucose rise is about the same. So this is an indication that there's also an impairment of beta cell function. What about the intervention protocol? The intervention protocol, we did not find a difference between the aligned and the misaligned state. And again, when I say misaligned, here's the sleep-wake cycle and the rest activity cycle is misaligned. But the eating-fasting cycle is aligned with the central clock. And so, so based on this paper, so just to summarize, we've shown that with this intervention protocol based on meal timing, we can prevent the internal misalignment between the endogenous circadian glucose and core body temperature rhythm. We can prevent the adverse effects on glucose control and pancreatic beta cell function. Just yesterday, I briefly showed some preliminary data. We're still working on that, on cardiovascular risk markers. And we've also published on anxiety-like and depression-like mood that can be corrected. And interestingly, the mood effects and the glucose tolerance effect correlate with the degree of internal circadian misalignment. So the more circadian misalignment people are, the worse their glucose tolerance, the worse their anxiety and depressive-like mood. So to summarize, so late eating per se, while clamping as much as well as we could all the other factors involved, increase the drive for food intake, decreases the amount of calories burned, and changes adipose molecular pathways towards growth. The concurrence of melatonin together with high glycemic load, as many of you may have heard before, is bad, but it's especially bad if you are a carrier of the MTNO1B risk SNP. So circadian misalignment has a lot of adverse effects, and I've just summarized those. But I think really exciting that there a promise for meal timing as a potential countermeasure against such adverse effects of misalignment that may be also of relevance to shift workers. And so just the final kind of take home or food for thought, I guess, is some gaps and opportunities that I see. I think, when I say onion, I'm thinking about unpeeling the onion. So if you come across or are doing chrononutrition studies, think about the many aspects. And so Joe Takahashi, as many of you have seen, recently published this beautiful study on wrapping that onion, so looking at calorie restriction separate from fasting duration, separate from meal timing. So I think that's really important for the field to really unravel the contributing factors and their potential interactions. I think also when changing either exercise timing or food timing or light timing, we should think about the effect it may have on other factors that may impact your outcome, so that we either control for it or at least document and record it. I think for meal timing, I think really important to think as a field for, are we using the clock on the wall? Are we using a gold standard circadian phase marker? Or are we using a proxy measure that might be more easily scalable, such as the habitual sleep-wake cycle? I think we have to think about many other aspects, such as differences based on our genetics towards personalized medicine, disease states, sex differences, race, ethnicity, work schedules, and of course, all of these may be different depending on the outcome variable you're looking at. And that is basically true for the whole list above. But the take home is, I think we're in an exciting time now with promise for the use of chrononutrition in the broadest sense for prevention and treatment approaches. So with that, I would like to thank all my collaborators and you for your attention and for the great honor. Thank you. And I'll leave you with this slide to wonder what this is. And maybe some of you will see it, but it's difficult to see. So, and as you see, the only contribution I have is to cheer them on. And we are looking for postdocs, and I'm happy to take any questions. Yes, thanks.
Thanks, Frank, for always another very nice presentation. Were they eating exactly at the same time as they ate during their day schedule?
Yes, yes, they were. Let me just go back to that. So, so as you see on the first two days in the laboratory, and then on day 5, 6, so after the constant routine, they have a 12-hour recovery sleep episode and then 2 more recovery days. And so the meal timing is kept constant if you look at the bottom protocol. Yeah.
So as a consequence, then this will, let's say, lead to sleep fragmentation, which is frequent in night workers. But what was your experience? What they felt during the night? Were they hungry? What were they doing?
Right, right. Yeah, so one comment to just make here that I didn't point out, but as you see, both protocols have scheduled 1-hour wake episodes in dim light that were done specifically such that we could actually make this happen, because otherwise how are you going to give them food intake during the biological day when they're actually sleeping during the biological day. So this is really, from that point of view, a proof of principle study. The next step now is to translate this into a protocol that can actually be married with a true exposure of a shift worker. So yes, we're looking into the factors related to hunger and appetite, but so they did report, and this is I don't think I've even seen the graph, but I believe that they did report increased hunger for the overnight times in the intervention protocol, because they have extended wakefulness without food intake. So again, so that that would need to be taken into account when translating. So how do you carry people over during an extended overnight work shift?
Because I'm afraid if you keep a real shift worker unfed all night, they're going to eat a lot when they come back in the morning, maybe before going to bed.
Right, yeah. Yeah, so absolutely true, Dan. So, I think that's a tricky thing. And so once you start to think about those solutions, there are many directions one can go. And one could either choose the mechanistic route and tease them out one by one, or one can use a more translational approach and combine a lot of what we know about nutrition to help them cope in that across the night work. Yeah. Yes.
You might have reported it, but are your circadian phase markers more stable in the individuals of a meal intervention, or the eating times aren't changing? So things like
it seems that they are equally stable. So if we look at the change graphs on the bottom, and if we look at the core body temperature, it's about, I think it's in the order of 35 to 40 minutes or so that they drift across the protocol. So yeah, I don't think we found differences in the degree of which the drift was not significantly different.
Yeah. Oh, okay. Yeah. So the fee schedule is not in training.
Right. Yeah.
I'm just wondering, to what extent controlling eating time in people that are traveling, where all of the time things are changing, whether you can nudge, or people that want to shift their clock and they're rotating the night shift, whether you can nudge their physiology using meal timing.
Right.
Assuming that's something you're going to look at some point.
Yeah, so at least in this protocol design, there was no effect of when the meals were occurring, which is not to say that it couldn't happen if designed maybe in a different way. It could be that because the meals were spread out more, maybe if we would consolidate the window to a narrower window, we're going to get a bigger Zeitgeber strength, then maybe there are effects visible. Yes, go ahead.
Yeah, so I'm really impressed by your work. So just a kind of very provocative idea. So when we think about that the obesity rate increased so dramatically over the last 30, 40 years, is it possible that there could be a correlation in the timing of meal, that people tend to, over the last 30, 40 years, tend to eat later in the day, and that could be the major driver?
Right, right. Yeah.
What's your view?
Yeah, no, exactly. I think that's a great idea, and kind of in the back of my mind, those ideas are. But the tricky thing is there is no good evidence. There's no good data available in large-scale populations across the decades on meal timing. So people have been tracking what they eat very well, or relatively well, but not when they eat. So this is really something, I think, a gap in our understanding. But I agree with you. It would be otherwise a perfect question to ask. Yes.
This may seem like an odd question, but I appreciate there's no calories in water, but were your participants able to have water at night or day?
Good question. Yeah. So we gave them 2.5 liters per 24-hour, equated to 24-hour time point. So we did allow them to request additional water. We would give them 100 ml. We would annotate this. But the amount of water they would request on top of it is fairly limited. But it's a good point in the sense that it could. And so the other thing is we do not allow them to drink water before test meals, because otherwise we may dilute, extend the stomach, dilute. So we don't think it's a big factor, but it's an important point. And I have to go back because we did look at thirst control. I don't think we found a circadian rhythm in thirst in our typical protocols. We did find, and I didn't mention it, a very strong circadian rhythm in nausea, with a peak in the biological morning, and some of you may relate to that. I think we had a question. Yeah.
So thinking more about translating this into practice, for a third shift worker, and a lot of times of the year their commute home would be bright light, and so would that change some of the timing they were exposed to this big bolus of bright light right before the rest phase?
Yeah, so one of the important factors of this, and somebody said it before as well, is that one consequence of the morning commute that night workers have is it locks their circadian phase such that they can't really adapt to night work very easily. And secondly, also the fact that most night workers revert back to sleeping at night and being awake during the daytime on free days to be with friends and family adds to that. And indeed, if you look at the literature, there's general agreement that unless you're working on oil rigs or Antarctic stations, your circadian rhythm as a night worker is typically, your biological night is still in the night. But that might not be a question. Is your question whether the light exposure would influence
Meal timing.
meal timing?
Yeah.
Well, the meal timing was locked in, but you mean the effect of meal timing?
Would it change when the ideal meal timing would be? Since in your protocol they were exposed to constant dim light, but a third shift worker would get a big hit of bright light before at the end of their shift.
Right. So if the, and this is pure speculation because we haven't done it, but if the central clock would be shifted, let's say you give a big bolus of light in the morning hours and you get a phase advance, then if it's substantial and you still keep your eating fasting cycle on a 24-hour day, then it may not. Then in fact your dinner might be into the biological night already. So it's really, in my mind at least, and again we haven't done these studies, it's the concurrence of eating with either the biological night or the later circadian evening, where it doesn't seem to bode well. We're trying to and of course, this is the other perspective, right? So we're living in an obesogenic environment, but one could say why has this system evolved? It may actually help us to cope with the overnight fast, both the glucose intolerance, which is helpful to not get hyperglycemia, as well as mechanisms that would facilitate storage of energy So it's not all bad, it's just in this time and age that. Thank you for all the questions.
Thank you so much, Frank.